let us see about cloning vectors. Class 12 Biotechnology, under that cloning vectors. So let us define cloning vectors. Any DNA molecule that is responsible for carrying of the desired gene that needs to be inserted into the host organism are known as cloning vectors. Okay, so that is the definition. Any DNA molecule. Okay, it's a DNA molecule. It carries desired gene. Okay, and it helps in transferring that desired gene into the host organism. Okay, so it is any DNA molecule. It carries desired gene. It helps in transfer of that desired gene into the host organism. Example, plasmid. So you all know what is plasmid. So plasmid is extra chromosomal extra chromosomal autonomously replicating genetic content. Okay, extra chromosomal autonomously replicating genetic content. Okay, it is seen in bacterial cells. It is present in bacterial cells other than the normal chromosomal DNA. Already in bacterial cell there is a nucleus and the genetic material. Other than that, it is something extra present in the bacterial cells. Okay, so extra chromosomal autonomously replicating genetic content is called plasmid. Okay, and this plasmid will help in transfer of the desired gene into the host organism. This plasmid will help in the transfer of desired gene into the host organism. And plasmid will contain two important things. Okay, number one, ORI. ORI means origin of replication. Origin of replication. Okay, so you know that DNA replicates. Okay, this particular site can be called as RI. So RI is the site of replication as the new desired gene enters the host organism. So there it is the site of replication as the new desired gene enters the host organism. So this is number one, and number two is ARG. That is nothing but antibiotic resistant gene. Antibiotic resistance gene. So a plasmid will contain two things. One is RA, origin of replication, and number two, antibiotic resistant gene. Okay. Now I am going to introduce you something called a very commonly used plasmid. A commonly used plasmid, that is okay. About this we will see now. Okay. Okay, so we are going to see about the commonly used plasmid. Okay, the most commonly used plasmid, that is PBr322. So let us see about what is this PBr322. Many of you may wonder what is this letters and numbers. So I'm going to give an introduction about what is this PBr322. Okay, so it's a commonly used cloning vector, especially in a bacteria called E. coli. So commonly used cloning vector in E. coli. It was first introduced in the year 1977. Okay, first introduced in the year 1977. Small p, this p stands for plasmid. So you all know plasmid means extra chromosomal, autonomously replicating genetic content. So P stands for plasmid. BR refers to the names of the scientist. B is Bolivar, okay, a scientist, and R for Rodriguez. So these people introduced this particular cloning vector, Bolivar and Rodriguez. And 322 refers to the order of sequence in which it has been derived, order of sequence. Okay, and it is in 4361 base pair of the length of double-stranded DNA. So you know that DNA is a polynucleotide chain. It has many base pairs, and this particular thing is 4361 base pair. DS means double-stranded DNA. So in that length it comes. It has two antibiotic resistance, ampicillin and tetracycline. That is what written here as AMR and tetracycline. Okay. So AMP and TET refers to ampicillin and tetracycline antibiotic resistant genes. And if you wonder what are these, okay, ECORP1, HIND3, BAMH1, SIL. So these are restriction sites, nothing but restriction sites. There are restriction enzymes and they are called restriction sites. Okay, and especially this antibiotic resistant genes are the selectable markers. And ROP refers to, it encodes for restrictor of plasmid that codes for a protein. Okay, it codes for a protein, ROP. Okay. So this is what the description of PBr322. Okay, commonly used cloning vector, first found in the year 1977. Okay, P stands for plasmid, BR stands for scientist name, Bolivar and Rodriguez. 322 refers to the order of sequence in which it has been derived. And if you take the length of the DNA, it gets placed in 4006 361 pair, base pair in the length of the double-stranded DNA. It has two antibiotic resistant genes, ampicillin and tetracycline, and ROP codes for a protein. So I have introduced this. In my next video, I will explain the features of cloning vectors. Hope you enjoyed this video. Thank you. Kindly like, share and subscribe.